The structure of the nucleosome core particle was determined after first isolating nucleosomes from the unfolded chromatin by digestion with particular enzymes, called nucleases, that break down DNA by cutting between the nucleotides. After digestion for a short period, only the exposed DNA between the core particles, the linker DNA, is degraded. An individual nucleosome core particle consists of a complex of 8 histone proteins. 2 molecules each of histones, H2A, H2B, H3, and H4, and a double-stranded DNA, 147 nucleotide pairs long, that winds around the histone octamer. Figure 522. The high-resolution structure of the nucleosome core particle was solved in 1997, revealing in atomic detail the disc-shaped histone complex around which the DNA is tightly wrapped, making 1.7 turns in a left-handed coil. Figure 523. The linker DNA between each nucleosome core particle can vary in length from a few nucleotide pairs up to about 80. The term nucleosome technically refers to a nucleosome core particle plus one of its adjacent DNA linkers. See Figure 522, but it is often used to mean just a nucleosome core particle. The formation of nucleosomes converts a DNA molecule into a chromatin thread approximately one-third of its initial length, and it provides the first level of DNA packing. All 4 of the histones that make up the nucleosome core are relatively small proteins with a high proportion of positively charged amino acids, lysine and arginine. The positive charges help the histones bind tightly to the negatively charged sugar phosphate backbone of DNA. These numerous interactions explain in part why DNA of virtually any sequence can bind to a histone core. Each of the core histones also has a long N-terminal amino acid tail, which extends out from the nucleosome core particle. See Figure 523. These histone tails are subject to several types of covalent chemical modifications that control many aspects of chromatin structure. The histones that form the nucleosome core are among the most highly conserved of all known eukaryotic proteins. There are only 2 differences between the amino acid sequences of histone H4 from peas and cows, for example. This extreme evolutionary conservation reflects the vital role of histones in controlling eukaryotic chromosome structure. Recently, histones have also been found in archaea, prokaryotes that form a phylogenetic kingdom distinct from eukaryotes and bacteria, discussed in Chapter 1. Chromosome packing occurs on multiple levels. Although long strings of nucleosomes form on most chromosomal DNA, chromatin in the living cell rarely adopts the extended beads on a string form seen in Figure 521b. Instead, the nucleosomes are further packed upon one another to generate a more compact structure, the 30 nanometer fiber. See Figure 521a. Packing of nucleosomes into the 30 nanometer fiber depends on a 5th histone called histone H1, which is thought to pull the nucleosomes together into a regular repeating array. This linker histone changes the path that DNA takes as it exits the nucleosome core, allowing it to form a more compact structure. Figure 524. The 30 nanometer fiber that results is illustrated in Movie 5.2 and in Figure 525, which provides an overview of the various levels of chromosome packing. We know that the 30 nanometer chromatin fiber can be compacted still further. We saw earlier in this chapter that during mitosis, chromatin becomes so highly condensed that the chromosomes can be seen under the light microscope. How is the 30 nanometer fiber folded to produce mitotic chromosomes? The answer is not yet known in detail, but it is known that the 30 nanometer fiber is folded into a series of loops, and that these loops are further condensed to produce the interface chromosome. Finally, this compact string of loops is thought to undergo at least one more level of packing to form the mitotic chromosome. Figure 526, and see Figure 525. The regulation of chromosome structure. So far, we have discussed how DNA is packed carefully and tightly into chromatin. We now turn to the question of how this packaging can be dynamic, allowing rapid access to the underlying DNA. The DNA in cells carries enormous amounts of coded information, and cells must be able to get to this information as needed. In this section, we discuss how a cell can alter its chromatin structure to expose localized regions of DNA and allow access to specific proteins, particularly those involved in gene expression and in DNA replication and repair. We then discuss how chromatin structure is established and maintained, and how a cell can pass on some forms of its structure to its descendants. The regulation and inheritance of chromatin structure play an important part in the development and growth of eukaryotic organisms. Changes in nucleosome structure allow access to DNA. Eukaryotic cells have several ways to adjust the local structure of their chromatin rapidly. One way takes advantage of chromatin remodeling complexes, protein machines that use the energy of ATP hydrolysis to change the position of the DNA wrapped around nucleosomes. By pushing on the tightly bound DNA as they move along, these complexes can loosen, decondense the underlying DNA, making it more accessible to other proteins in the cell. Figure 527. During mitosis, at least some of the chromatin remodeling complexes are inactivated, which may help mitotic chromosomes maintain their tightly packed structure. Another way of altering chromatin structure relies on the reversible chemical modification of the histones. The tails of all four of the core histones are particularly subject to these covalent modifications. For example, acetyl phosphate or methyl groups can be added to and removed from the assembled nucleosome by enzymes that reside in the nucleus. These modifications of the histone tails have little direct effect on the stability of an individual nucleosome, but some seem to directly affect the stability of the 30 nanometer chromatin fiber and the higher order structures discussed earlier. Most importantly, these modifications affect the ability of the histone tails to bind specific proteins and thereby recruit them to particular stretches of chromatin. Different patterns of histone tail modifications attract different proteins, some of which cause further condensation of the chromatin, whereas others facilitate access to the DNA by decondensing chromatin. Specific combinations of tail modifications and the proteins that bind to them have different meanings for the cell. For example, one pattern might indicate that a particular stretch of chromatin has been newly replicated, whereas another indicates that the genes in that chromatin should be expressed. Figure 528. Like the chromatin remodeling complexes, the enzymes that modify histone tails are tightly regulated. They are brought to a particular region of chromatin by other cues, particularly by interactions with proteins that bind to specific sequences in DNA. We discussed these proteins in Chapter 8. The histone-modifying enzymes work in concert with the chromatin remodeling complexes to condense or decondense stretches of chromatin, allowing local chromatin structure to change rapidly according to the needs of the cell. Interphase chromosomes contain both condensed and more extended forms of chromatin. The localized alteration of chromatin packing by remodeling complexes and histone modification has important effects on the large-scale structure of interphase chromosomes. The chromatin in these chromosomes is not uniformly packed. Instead, regions of the chromosome that contain genes that are being expressed are generally more extended, while those that contain quiescent genes are more compact. Thus, the detailed structure of an interphase chromosome can differ from one cell type to the next, helping to determine which genes are expressed. The most highly condensed form of interphase chromatin is called heterochromatin, from the Greek heteros, different, plus chromatin. It was first observed under the light microscope in the 1930s as discrete, strongly staining regions within the mass of chromatin. Heterochromatin typically makes up about 10% of an interphase chromosome, and in mammalian chromosomes it is concentrated around the centromere region and in the telomeres at the end of the chromosomes. The formation of the most common form of heterochromatin is induced by a particular set of histone tail modifications, including the methylation of lysine residue 9 in histone H3. See Figure 528. These modifications attract a set of heterochromatin-specific proteins, which then induce the same histone tail modifications in adjacent nucleosomes. The new tail modifications in turn recruit the same set of heterochromatin-specific proteins, causing a spreading wave of condensed chromatin to propagate along the chromosome. In this manner, an extended region of heterochromatin is established along the DNA. Most DNA that is permanently folded into heterochromatin in the cell does not contain genes. Because heterochromatin is so compact, genes that accidentally become packaged into heterochromatin usually fail to be expressed. Figure 529. Such inappropriate packaging of genes in heterochromatin can cause disease. In humans, the gene that encodes beta-globin, which forms part of the oxygen-carrying hemoglobin molecule, is situated next to a region of condensed chromatin. If, because of an inherited DNA deletion, that region of heterochromatin spreads, the beta-globin gene is poorly expressed and the person develops a severe form of anemia. Perhaps the most striking example of the use of heterochromatin to keep genes shut down or silenced is found in the interphase X chromosomes of female mammals. Female cells contain 2 X chromosomes, where male cells contain 1 X and 1 Y. Because a double dose of X chromosome products would be lethal, female mammals have evolved the means of permanently inactivating one of the 2 X chromosomes in each cell. At random, one or other of the 2 X chromosomes in each cell becomes highly condensed into heterochromatin early in embryonic development. Thereafter, the condensed and inactive state of that X chromosome is inherited in all of the many descendants of those cells. Figure 530. The rest of the interphase chromatin is called euchromatin, from the Greek eu, true, or normal, plus chromatin. Although we use the term euchromatin to refer to chromatin that exists in a more extended state than heterochromatin, it is now clear that both euchromatin and heterochromatin are composed of mixtures of different chromatin structures, each established and maintained by different sets of histone tail modifications that attract distinct sets of non-histone proteins. Figure 531. Changes in chromatin structure can be inherited. As we have just seen, certain types of chromatin structure can be passed from a cell to its descendants. The progeny of a cell in which the maternal copy of the X chromosome is condensed and inactivated, for example, will also condense and inactivate their maternal X chromosome. How is such inheritance of chromatin structure possible? When a cell replicates its genome, each daughter DNA helix receives half of its parent's histone proteins. With those histone proteins come the covalent modifications associated with the type of chromatin structure that was present in each particular region of that parental chromosome. Thus, each daughter chromosome will initially contain an intermixed set of 2 types of nucleosomes, those that contain the modified histones inherited from its parent chromosome, and those that contain newly synthesized histones which have not yet been modified. At this point, proteins that recognize the modified histones may bind to the parental histones and deposit the type of modification on the nearby virgin histones, re-establishing the pattern of chromatin structure found in the parent. Figure 532. The ability to inherit localized chromatin structure helps eukaryotic cells to remember whether a gene was active in its parental cell, a process that appears to be critical for the establishment and maintenance of different cell types, tissues, and organs during the development and growth of a complex multicellular organism. This type of inheritance does not involve passing along specific DNA sequences from one cell generation to the next, but instead depends on passing along specifically modified histone proteins. It is an example of epigenetic inheritance, from the Greek epi, on, because it is superimposed on genetic inheritance based on DNA. Other forms of epigenetic inheritance are discussed in Chapter 8. Essential concepts. Life depends on the stable and compact storage of genetic information. Genetic information is carried by very long DNA molecules and encoded in the linear sequence of nucleotides, A, T, G, and C. Each molecule of DNA is a double helix composed of a pair of complementary strands of nucleotides held together by hydrogen bonds between GC and AT base pairs. A strand of DNA has a chemical polarity due to the linkage of alternating sugars and phosphates in its backbone. The two strands of the DNA double helix run antiparallel, that is, in opposite orientations. The genetic material of a eukaryotic cell is contained in a set of chromosomes, each formed from a single, enormously long DNA molecule that contains many genes. When a protein-coding gene is expressed, part of its nucleotide sequence is copied into RNA, which then directs the synthesis of a specific protein. The DNA that forms each eukaryotic chromosome contains, in addition to genes, many replication origins, one centromere and two telomeres. These sequences ensure that the chromosome can be replicated efficiently and passed on to daughter cells. Chromosomes in eukaryotic cells consist of DNA tightly bound to a mass of specialized proteins. These proteins hold the DNA into a compact form. The complex of DNA and protein in chromosomes is called chromatin. The most abundant chromosomal proteins are the histones, which pack DNA into a repeating array of DNA protein particles called nucleosomes. Nucleosomes pack together with the aid of histone H1 molecules to form a 30 nanometer fiber. This fiber is generally coiled and folded, producing more compact chromatin structures. Chromatin structure is dynamic. By temporarily decondensing its structure, using chromatin remodeling complexes and enzymes that covalently modify histone tails, the cell can ensure that proteins involved in gene expression, replication, and repair have rapid localized access to the necessary DNA sequences. Some forms of chromatin have a pattern of histone tail modification that causes the DNA to become so highly compacted that the packaged genes cannot be expressed to produce RNA and protein. Chromatin structure can be transmitted from one cell generation to the next, producing a form of epigenetic inheritance that helps the cell to remember the state of gene expression in its parent cell.